Dear colleagues, let me express my gratitude to the organizing committee for inviting me to speak at the International Cancer Conference. As you may have noticed, the title of my presentation is somewhat intriguing, though it is not a market employ. Instead, it is an urgent need for a new scientifically grounded perspective on the cancer problem. Over the past 25 years, our experience in the interdisciplinary clinical research in oncology has enabled us to accumulate valuable information and share it with you. The global scientific community in oncology possesses vast resources. Indeed, much is known about the cancer biology and the molecular genetic mechanisms of its occurrence. Certain achievements have been made in cancer treatment. However, global result and long-term outlook for morbidity and mortality remain highly unfavorable. By 2050, the world economy will experience damage that will amount to more than $25 trillion. Why is the cancer process uncontrollable? The scientific response is solely based on cancer's genetic characteristics, and it is considered a disease of genes. This viewpoint comes from the perspectives of the molecular genetic level of organization of living matter. Let us re-examine the levels of organization of living matter, which can help us bring light to the dark dungeon of the cancer problem. Pay attention to the molecular genetic level, which represents the boundary between the living and the non-living matter. Exactly, at this level modern scientific attention is directed in the study of the cancer issue. The primary resources dedicated to cancer research are also directed towards this level, considering cancer a genetic disease. But cancer is a global problem. It is a problem of civilization and the human population. Therefore, consideration of the cancer problem must begin at the population level. What unfavorable factor can affect a person at the population level? It is chronic stress. Chronic stress gained momentum in the 20th century and has since become a global epidemic of the 21st century, a significant issue for modern civilization. The accumulated extensive scientific data shows that chronic stress permeates all levels of the human body and has a destructive effect on it. Specifically, at the organismal level, chronic stress has a destructive effect, first of all on the nervous system, and the main integrative, control, and regulatory system of the body, leading to extremely negative consequences for the organism as a whole. A body thermographic map of emotions is a straightforward and clear way to qualitative evaluation of the impact of human emotions on the body. Compared to feelings of happiness, depression appears terrible. Behind this unsightly blueness is hidden a pronounced violation of capillary blood supply and the ischemia of body tissues, which underlie accelerated biological aging and numerous chronic diseases, including diseases of civilization. So, the main feature of the influence of chronic stress at the organismal level is the disruption of the nervous regulation of the organism. Major disruptions occur in the central nervous system, particularly in the brain, which we refer to as the brain disintegration syndrome. In essence, these disruptions indicate that the organism begins to lose its cohesion as a unified whole. The brain disintegration syndrome includes dissociated changes in four major brain systems, decreased activity and dysfunction of the prefrontal cortex and hippocampus, stagnant excitation in the suprasegmental vegetative structures of the limbic reticular complex, a shift in the vegetative balance towards a state of hyposympathetic atonia, with descending tonic signals flowing to the tissues and target organs of the body. Let me remind you that the brain is the key organ regulating the processes of cyanogenesis, a complex of protective and adaptive mechanisms aimed at self-regulation, self-restoration, self-healing. Brain disintegration syndrome impedes the full implementation of cyanogenesis processes and in clinical practice significantly complicates patient treatment. The processes of cyanogenesis, regulation by the central nervous system and the brain, are essentially the opposite of those of pathogenesis. Therefore, in modern treatment of almost every patient, both pathogenetically oriented and cyanogenetically oriented therapies should be used. In other words, without effective therapeutic interventions on the central nervous system and higher nervous activity, it is extremely difficult to achieve recovery or stable remission of chronic disease. This is fully applicable to the treatment of cancer patients. This is the approach we use in our clinical work. In clinical practice, the brain disintegration syndrome resulting from chronic stress always manifests itself in mental, behavioral, and vegetative disorders. Indeed, anxiety and depressive disorders, along with corresponding behavioral disorders, have also currently acquired the character of global epidemic with a poor long-term prognosis. Despite the significant role that stress-related mental disorders play in the pathogenesis of diseases, their rigorous scientific assessment remains impossible due to the inherent, irreducible, irremovable subjectivity. That is, all existing instrumental methods of brain research and neuroimaging are meaningless in diagnosing the psycho-emotional manifestations of chronic stress, not to mention standard psychometric methods. At the same time, at the organismal level, there is an opportunity for a strictly objective assessment of the functional state of the entire organism. We believe that for now the only possible method is the analysis of heart rate variability. Since 1960, the USSR had been developing the heart rate variability analysis technique to assess astronauts' functional state quickly and reliably for space medicine purposes. This technique has been continuously improved in various countries around the world, and now it is integrated into many devices for electrocardiography and Holter monitors. For a long time, the heart rate variability analysis technique had limitations due to recording errors and, consequently, difficulties in interpreting results, especially in the presence of arrhythmias and other heart abnormalities. Professor Vladimir Yemelyanenko managed to eliminate these shortcomings, modify the technique, and get the opportunity not only to accurately determine the vegetative imbalance in the body and the level of hyposympathetic atonia, but also to calculate the so-called E-index, a health index that reflects the coherence of integrative processes in the body. Studying the index dynamically provides an objective, comprehensive assessment of the functional state of the human body and allows for the evaluation of the effectiveness of therapeutic interventions on the patient's body, including cancer patients. For instance, using a mouse model, Professor Erika Sloan convincingly demonstrated that vegetative imbalance in the form of hyposympathetic atonia induced by chronic stress leads to a 30-fold increase in spontaneous metastasis for primary breast tumor cells to distant tissues. In our opinion, hyposympathetic atonia is a sort of red button for the organism which in cancer patients should always be deactivated in order to achieve control over the cancer process. Frankly speaking, this is quite challenging to do. At the lower levels of the human body, organ and tissue, disturbances of nervous regulation under the influence of chronic stress manifest themselves as specific organic changes, collectively known as the disease of civilization. Cancer also belongs to this category of diseases. At these levels, diagnostic methods for objectifying organic changes in organs and tissues are widely used in modern oncology. The slide presents scientifically proven changes at the cellular and molecular genetic levels affected by chronic stress. The all-pervasive damaging effect of chronic stress on the body is demonstrated through various mechanisms associated with stress hormones and hyposympathetic atonia. It is important to know that a number of pathological changes at the molecular, genetic, cellular, and tissue levels are directly related to carcinogenesis. In other words, against the background of chronic stress, the body has everything for the occurrence, development, and progression of cancer. In particular, tissue ischemia, persistent inflammation, endogenous carcinogens, and critical DNA damage occur, the activity of antitumor immunity is suppressed, and other changes take place. Carcinogenesis without exogenous carcinogens is entirely plausible. Stressful carcinogenesis. We reported this in our article published in the Journal Medical Hypothesis in 2009. The National Cancer Act, also known as the War on Cancer, signed by President Richard Nixon, played a significant role in fundamental research in cancer problem. To a certain extent, he gave a significant impetus to scientific research in the global oncology. However, nowadays the scientists' focus has shifted towards the molecular genetic level of the human body, in fact, to the body between living and non-living. For example, the world's largest and most comprehensive resource on genetic mutations associated with human cancer was created, the Catalog of Somatic Mutations in Cancer, COSMIC. As you can see, at this lowest level of the human body, the number of variables that potentially related to cancer is vast. And this is not static data. It has been updated every 3-4 months for the past 20 years. The future victory in the war on cancer is associated exactly with the individual gene-oriented approach to the treatment of cancer patients. As shown in the slide, the lower the level of the human organism under consideration, the greater the variety of possible changes that researchers encounter. All processes at the molecular, genetic, and cellular levels occur exclusively locally, manifested through intercellular and intermolecular interactions. To trigger systemic reactions of the organism or to carry out effective systemic treatment of cancer patients, the involvement of higher levels of the organism is required. Conclusions based solely on the molecular, genetic, and cellular levels do not allow, for example, to explain the cancer paradox known as Petau's paradox. The paradox lies in the fact that there is no direct correlation between the size of an organism and its risk of developing cancer, although each living cell theoretically has an equal probability of degenerating into a cancer cell. Therefore, larger animals should supposedly suffer from cancer more often than smaller animals. However, this is not the case. From the standpoint of the organismal level, each animal, regardless of size, functions as a single established organism, meaning the number of cells is irrelevant. Nervous regulation, integrating the work of cells, tissues, and organs into a single organism, is of decisive importance. It's time to shed light on a system error in cancer research and treatment. The system error in cancer research and treatment is neglecting the organismal level of experimental animals and cancer patients, in essence, ignoring the participation of the central nervous system in the cancer process. To put it succinctly, it is impossible to see the whole organism of a suffering cancer patient by focusing solely on their cells, molecules, and genes. This is comparable to a detailed study of broken bricks, in the piles of which one tries to see a huge house, with life support systems, apartment, interiors, and residence. Let us say once again that a kind of red button of the organism is a persistent imbalance in the autonomic nervous system, resulting in the state of hypersympathetic atonia. This pathological condition accompanies the cancer process at all stages and is accessible to objective control. All available pharmacological and non-pharmacological methods in the medical arsenal that can eliminate hypersympathetic atonia. In essence, activating the body's green button and achieving a state of normatonia. Right now, this will significantly improve the effectiveness of any existing cancer treatment methods. We first presented this clinical possibility of controlled cancer regression in a video report at the International Conference on Biomedical and Cancer Research in Tokyo in October 2019. We hope that the content of this presentation will help rethink the approach to global problem of cancer. Let us use the human brain in the global war against cancer in the 21st century. Thank you very much for your attention.